Hi, everyone. Thanks for joining us in Auditorium 2. If you wanted to come and learn all about how mitochondrial changes in FAODs and understanding strange words like ROS or ROS and cardiolipin, you're in the right place. I just want to remind everyone that if you have questions, please go ahead and put those in the Q&A section on your screen, and we will try to get to those as fast as we can at the end of the presentation. And if we can't get all of your questions answered, we will try to get those answered and back to you throughout the conference. Joining us for our very first time today, we'd like to have Dr. Eric Goetzman here as our speaker. Following his undergraduate studies at the University of Minnesota Morris, Dr. Goetzman obtained a PhD in medical genetics from the University of Alabama, Birmingham, where he investigated fatty acid and metabolism and rare metabolic disorders. After completing a postdoc training at the Mayo Clinic and UPMC Children's Hospital of Pittsburgh, he joined the research faculty of the University of Pittsburgh School of Medicine in 2006. He is currently an associate professor of pediatrics, where he continues to research various aspects of energy metabolism, mitochondrial function, and rare metabolic diseases. Please join me today in welcoming Dr. Goetzman.
Hello. Thank you for that great introduction, Stephanie. So what I wanted to talk to you about today is how the mitochondria change within FAODs. And as Stephanie said, we're going to talk about these strange words, ROS and cardiolipin. She outlined my background pretty well, but I just wanted to highlight a couple of key points about my role in studying fatty acid oxidation over the last 20 plus years. I did my graduate training at the University of Alabama, Birmingham, in the lab of Phil Wood. And in Phil's lab, we developed over many years several mouse models of FAODs that are still in use today. These are the SCAD, MCAD, LCHAD, VLCAD, and CPT1 deficient mouse strains, which are all, as I mentioned, still in use today for studying these disorders and new therapies. After I left Phil's lab, I joined Jerry Vockley's lab. You probably all know Jerry by now. I know he spoke yesterday and again this morning, I believe. And in Jerry's lab, I learned more about the human side of these disorders. I learned about the enzymology, how to analyze patient cells, and a lot about biochemistry. So I don't expect you to be able to read this slide at all. What this is a map of all of the metabolic pathways that are currently going on in your cells and how they all intersect and influence each other. I don't want to, I'm sure you heard what I said. The visuals aren't all that important. So we'll get back to where I was. So we developed mouse models. This was a map of all of the metabolic pathways that are currently going on in your cells. And one of the interests in my research lab now is understanding how they all affect each other. If you disrupt one, like with a fatty acid oxidation disorder, what does that do to all of the other pathways that are interconnected with fatty acid oxidation? Today, we're going to talk about how the mitochondria change in FAODs. And I'm going to provide a little bit of data from recent papers showing how the general appearance of mitochondria, which we call morphology, changes in some patients, not all, with FAODs. And there's many factors that probably contribute to that, but we're going to stick to two of them that you may have heard of if you've been Googling or searching the web at all, and that you may be curious about, which are cardiolipin and reactive oxygen species, which we abbreviate as ROS or ROS. So as we go along, I'm going to present data from recent basic science research papers and show you how I think these two factors might play a role in FAODs. You've also, if you've ever Googled these disorders and read about mitochondria, you've probably seen this phrase before, that mitochondria are the powerhouse of the cell. They're like little batteries, and they provide the bulk of the energy, especially for organs that need a lot of energy, like the heart and muscle, particularly during exercise, and some other organs as well. Mitochondria are unique amongst organelles in that they have their own DNA, which encodes some of the genes for a group of complexes called the respiratory chain. If you can see my arrow, I'm showing the respiratory chain. They're conveniently numbered complexes 1 through 5. Some components of these are encoded in the mitochondria itself. And the mitochondria also has this unique double membrane, where the outer membrane is just sort of ordinary, but the inner membrane is highly convoluted and has all of these folds. And that serves to greatly increase the surface area of the inner mitochondrial membrane, which is where these key metabolic reactions are taking place, including the first steps, at least for fatty acid oxidation, the long chain enzymes. Carnitine palmitoyl transferase 1, or CPT1, is on the outside of the mitochondria. This brings fatty acids across this double membrane and hands them off to CPT2, which is stuck to the inside of the inner mitochondrial membrane, along with VLCAD, which is very long chain acyl-CoA dehydrogenase, and LCHAD. And some of the other enzymes, such as MCAD, will take on the fatty acids after they've been partially shortened. And these enzymes are believed to be in the matrix, which is this sort of goop inside the mitochondria, very rich in protein, where a lot of action is taking place with regards to metabolism. So as fatty acid oxidation produces electrons, it hands them off to the respiratory chain. They go down this respiratory chain and ultimately produce ATP, which in the mitochondrial version of a battery is the energy. So mitochondrial morphology or shape has been studied for quite some time. The first recorded observation of mitochondria goes back over 125 years to Richard Altman, who was looking through his microscope and noticing these little blips inside of the cells, and he drew pictures by hand of what he saw. In wing muscle, he drew these little black dots lined up within the fibers of the muscle tissue. Frog liver cells, he drew them as these red worm-like looking structures. And he didn't really know what they were, but he recognized that they were carrying out vital functions for the cell. Now we have much more sophisticated methods of imaging. We have microscopes that allow you to take very high resolution pictures, even videos, particularly in cultured cells where you can see easily into the cell and see what the mitochondria are doing. And so now we know that mitochondria actually come in many different shapes and sizes. And furthermore, they tend to form connected networks. They don't just exist in isolation most of the time, at least in cultured cells, which is where this has been intensely studied. So these pictures here at the top all represent individual mitochondria. And you can see, depending on the tissue, they could take on this kind of familiar looking kidney bean shape, which is how we tend to draw mitochondria. Or they could take on hairpin shapes or lattice structures, depending on the physical constraints of the cells that they're in. And as I mentioned, they're dynamic. They don't just sit there. They're moving around, they fuse, they divide. So over recent years, scientists have developed methods where in cultured cells, you can label an individual mitochondria. In this case, the scientist has labeled this mitochondria in green and the others in red. And then over time, you can watch to see, where does the green one go? Does it divide? Does it fuse? And what you see is that these things are constantly in motion, like little worms. They're just divided. Oh. So there it divides right there into two mitochondria, which then fuse with other mitochondria. And now you see the green signal is being spread out throughout the mitochondrial network. And these are other examples of similar phenomenon, labeling mitochondria and watching what they do. So we know a lot from this cultured cell technology that mitochondria are dynamic, that in human tissues we can't see inside, so we really don't know quite yet what's going on in the intact human body. So what do the mitochondria look like in FAODs? There's been a few recent papers that have used this imaging technology to look at the mitochondrial networks or in the shape of the mitochondria themselves in FAODs. Here on the left, we see at the top, these two pictures are from control subjects, and these are fibroblast cells. And here they've labeled the mitochondria with red, and this part that's outlined by the white dashed box is what you see here next to it. This is a zoomed in version of that box. And now they've used a computer to flip the red to white so you can really see the contrast. And what you see is these mitochondria form networks like I was just showing you in the video. And similar in this other control patient. In the LCHAD patients, on the other hand, they looked at several patients' cell lines in this paper. And what they saw was that most of them, but not all, show disorganized mitochondrial networks. And now you tend to have more mitochondria that are just individually sitting off by themselves in this sort of kidney bean morphology, rather than the elongated shape bound into the networks. Similarly, another paper has used heart cells and cultured heart cells, and then used a technique you've probably heard about in the news called CRISPR to knock out LCHAD. And what they saw was, when they used electron microscopy, which allows you to really zero in inside the cell, that the mitochondria in the control cells were mostly long with very easily discernible cristae inside. That's what we call those convoluted folds. Whereas the LCHAD knockout cells had they had some normal mitochondria. They tended to have much more of these round-shaped mitochondria with unusual looking cristae inside, more like a ring rather than the intricate folding pattern that was expected. So these papers would suggest that at least in some FAOD patients, there can be changes in mitochondrial morphology. And again, the disclaimer would be this is all done in cell culture. Similarly with VLCAD cultured cells, you can see on the left is a control cell. Mitochondria are mostly elongated. This blue arrow points to a long one. Some that probably just divided are these small circles. In the VLCAD patient cell line, you see the mitochondria look sort of empty inside, and the cristae are sort of like stripes rather than folds, and there's clearly something going on with the morphology in this patient. And again, I have to stress that we don't have evidence that this would be occurring in every patient, or certainly not in every cell type, as we don't really know yet what's going on in the human body. So if we just say that we're going to study this phenomenon, what would be some of the factors that could cause such a thing? Well, as I alluded to early on, my lab is greatly interested in this idea of a ripple effect. You disrupt something like FAOD, what happens to all of the other pathways around it? You end up with situations such as reduced energy production, less ATP. The fatty acids can't be broken down, so they accumulate, which has a lot of downstream effects. And then you also start to disrupt things like communication between the mitochondria and the nucleus, which is known to be poorly understood, but known to play a role in regulating how mitochondria divide, duplicate, etc. But the factors that we're going to talk about today are reactive oxygen species, which are known to be increased in many of these FAOD patient cell lines. And also we're going to look at changes in the membrane composition, something called cardiolipin. So first, let's get into cardiolipin. What is cardiolipin? The short answer would be cardiolipin is a phospholipid. So what's a phospholipid? Phospholipids are the building blocks of all of your cellular membranes. Phospholipids have a common structure. They all have what's called a hydrophilic head group. Hydrophilic means water loving. And then they have hydrophobic or water hating tails. The tails are just fatty acids that are stuck to the head group. So here immediately you probably see a link between phospholipids and fatty acid metabolism. So that these fatty acids are coming from somewhere and being synthesized onto these head groups. In the context of phospholipids, we call those fatty acids acyl chains that are stuck onto the head group. Now, the water-hating acyl chains try to escape the water, and so they tend to orient towards each other, and the head groups point to the outside. So what you get is this phospholipid sandwich, or a bilayer, as the scientific term would be. And this is the basic structure of all of your cellular membranes. Cardiolipin is a special phospholipid because it has 4 acyl chains instead of just 2, and it's found strictly in the mitochondrial membrane. So what does it do there? And it turns out we're just kind of scratching the tip of the iceberg in terms of understanding what cardiolipin does. But one of the things that's well established is that it helps to introduce curves in the inner mitochondrial membrane. Because it has 4 acyl chains, it takes up more space. And when you elbow a bunch of them together in one place, you get a curve, which is what allows these folds, which we call cristae, to form properly inside the mitochondria, to get that surface area, which is where all of the business of metabolism takes place. But cardiolipin also may be affecting many other aspects of mitochondrial biology. For example, it's thought that cardiolipin influences the formation of supercomplexes of the mitochondrial respiratory chain, those complexes that pass the electrons along and produce ATP. They don't like to exist just as single complexes floating around. It's more efficient for them to gather together in these complexes, particularly complexes 1, 3, and 4, like the glom onto each other, and that more efficiently can pass electrons and produce energy. It's also thought that cardiolipin, due to its role in membrane curvature, can play a role in how membranes fuse and split during mitochondrial dynamics. It clearly plays a role in the cristae, as I mentioned, causing these curves that are absolutely necessary for cristae formation. And lastly, cardiolipin might be playing a role in mitochondrial quality control, which is the process in which damaged mitochondria are sorted out from the functional ones and targeted for degradation. But we're primarily interested in fatty acid oxidation, so what does cardiolipin have to do with that? Well, it turns out that cardiolipin is a binding platform for the key enzymes of fatty acid oxidation, LCHAD, VLCAD, and CPT2. They all have domains in their protein structures that bind to cardiolipin. So this puts them all together in these curved areas of the inner mitochondrial membrane where they can efficiently channel incoming fatty acids from one enzyme to the next. And furthermore, Jerry Vakri's lab has shown that these complexes of FAO enzymes bind directly to the respiratory chain supercomplexes, which makes sense because now you can take the electrons that are coming from VLCAD and LCHAD and pass them directly into the respiratory chain and more quickly, more efficiently produce ATP. So some of the experimental evidence supporting a role for cardiolipin in fatty acid oxidation comes from some studies we did about 10 years ago with a patient mutation in VLCAD called A450P. VLCAD A450P variant enzyme, we found it had full enzymatic activity, but it could not bind to cardiolipin, which meant it was not in its correct place disrupting the channeling through this protein complex and making fatty acid oxidation inefficient and causing symptoms for the patient, even though the enzyme is actually fully functional. Other more recent evidence regarding CPT2, this paper just came out earlier this year, showed that CPT2 is very unstable and unhappy when it's not bound to cardiolipin. In this study, they purified the CPT2 enzyme and they studied its properties, and they saw that very quickly, in the matter of an hour, this black line that my arrow is on, the enzyme loses about half or more of its activity if there's no cardiolipin present. If they just add some cardiolipin to the tube with the protein, it's happy again. And now it can stay fully stable and functional over an hour, just sitting in this tube. And the more cardiolipin you add, it seems to be happier. The final piece of evidence comes from a disease called Barth syndrome, in which the patients have a defect in their cardiolipin. Another recent paper, I think this was also from 2021, compared the rates of fatty acid oxidation in control subjects, where the black bars, and Barth syndrome subjects, the gray bars. When the subjects were resting, the Barth syndrome patients had normal rates of fatty acid oxidation. But when you subjected them to a exercise stress, whereas the control subjects could double their fatty acid oxidation, the Barth syndrome patients could not, and now became energy deficient while exercising. Again, this is further evidence that cardiolipin plays a key role in promoting fatty acid oxidation. But it's more complicated than that. It turns out that FAO and cardiolipin are interdependent, and they rely upon each other for optimal function. So let's explain what I mean by that. So cardiolipin, for it to function at its best, has to have a certain composition of acyl chains, particularly in tissues that have high energy requirements, such as the heart. So when cardiolipin is made inside the mitochondria, it comes off the assembly line in what we call the immature form, where these acyl chains are just essentially random fatty acids. They could be 16 carbons long, 20 carbons long, could have 0 double bonds, 1 double bond, which is whenever you see this line, that indicates a double bond. And it can make a big difference because as you introduce double bonds, you get different structures of the acyl chains. You can get a kinked structure with 1 double bond, a hairpin structure with 2 double bonds. And we call these fatty acids unsaturated. You probably have seen that word, unsaturated fatty acids. If you take fish oil, you're doing that for your health because unsaturated fatty acids are deemed to be healthy. So that's probably a familiar term to you. So how does the cardiolipin get the acyl chains it wants? Because it turns out that each tissue has a preferred cardiolipin profile for its mature cardiolipin. Extreme examples would be brain, shown in this top panel, and heart in the bottom panel. Along the bottom here, all these numbers represent the individual cardiolipin species, where the total number of carbons in the 4 acyl chains is the first number, in this case 66, and then there's the colon, and the second number would be the number of double bonds present in the entire cardiolipin molecule, all 4 acyl chains. So it turns out the brain isn't picky at all. It's basically like, whatever acyl chains you got, I'll take, it's fine. There's many different cardiolipin species in the brain. Heart, on the other hand, which relies heavily on its mitochondria 24/7 for energy production, has a very specific cardiolipin it wants, which is this one, in which all 4 of the acyl chains have the same fatty acid, 18 carbons long with 2 double bonds. Another name for this is linoleic acid. And because of the double bonds, you get these hairpin structures and this unique crab-like shape to this cardiolipin species. So how each tissue gets its individual cardiolipin profile not at all understood. But what we think is going on is that there are two processes of cardiolipin remodeling. In the first process, as the immature cardiolipin comes off the assembly line into the matrix of the mitochondria, it has 4 random acyl chains. At this point, it can cross the intermitochondrial membrane, this phospholipid bilayer, into the space that's between the 2 membranes that the mitochondria has, the outer membrane and the inner. And there it encounters these enzymes called phospholipases, which strip off the acyl chains. And then an enzyme called TAZ, which has been fairly well studied because it turns out this is the enzyme that's mutated in Barth syndrome. TAZ steals acyl chains from other phospholipids in the membrane. How it knows which ones to select, we don't understand, but it steals these acyl chains and replaces the missing ones, and it keeps going until you get the desired species, the mature species of cardiolipin. Now, there's another remodeling pathway which is even much less well understood, in which the cardiolipin, through unidentified phospholipase in the matrix, loses acyl chain, and then an enzyme, which was for several years called MLCL-AT1, which is a mouthful, uses acyl-CoA. You might recognize that word. That is the substrate for VLCAD or MCAD, which are acyl-CoA dehydrogenases. It uses acyl-CoA as the fatty acid donor, and it remodels the cardiolipin. Now, what's really interesting is back in 2012, in this paper in a journal called PLOS ONE, Grant Hatch, a scientist at the University of Manitoba, his lab showed that MLCL-AT1 is really LCHAD. They just didn't know what it was, and so they gave it this name, but it turns out that it's really LCHAD. And so what this suggests is that LCHAD can play this auxiliary moonlighting role in the mitochondrial inner membrane, in which it's not only involved in fatty acid oxidation, but it may be playing a role in remodeling cardiolipin. Some evidence for this role of LCHAD comes from this paper by Al-Tibi et al., just came out earlier this year, in which they looked at the cardiolipin chain length and the number of double bonds in the acyl chains. And they used fibroblast cell lines from control subjects, VLCAD patients, LCHAD patients, and CPT2 patients. To make this data a little easier to digest, they've color-coded it so you can see the total number of carbons in all 4 of the chain lengths together, from 64 up to 86. Each has a color. The control fibroblasts, very similar overall to the VLCAD. And you see some modest changes in the LCHAD and CPT2 cell lines, in which they tend to have somewhat shorter acyl chains than the control. But where the differences really get interesting is when you look at the number of double bonds. In fibroblasts, it turns out that most of the cardiolipin acyl chains have 2 or fewer double bonds, which would be the green is 2, red is 1, blue is 0. VLCAD patients, this is shifted to the right to more double bonds. So the VLCAD patient cells have primarily 2, 3 is the purple, 4 is the blue, and 5 double bonds, orange, and even some 6 and 7. In LCHAD, it gets very interesting. Very few acyl chains with 0, 1, or 2. Instead, the bulk of the acyl chains have 3 double bonds, or 4, and some 5. CPT2, this is a little bit surprising, even more double bonds in the acyl chains. Now we're looking at 4, 5, and 6 double bonds. So as you disrupt the FAO pathway, clearly you disrupt the composition of the cardiolipin acyl chains. And furthermore, even if you go further upstream of these enzymes, if you look at an enzyme called ACSL1, which is the enzyme that puts this cofactor called CoA on the fatty acids, such that CPT1 can then import them into the mitochondria. What you see is the ACSL1 knockout mouse, which lacks this enzyme, also has reduced cardiolipin in its heart. Just focus on this first set of bars, which is that preferred cardiolipin in the heart with the 4 linoleic acids on it. The control mice are the white bar, the knockout mice are the black. Almost 5 times less of this cardiolipin species in the hearts of these mice. And furthermore, Grant Hatch's lab showed if you inhibit CPT1 with a chemical called etomoxer, which inhibits CPT1 irreversibly, you see a decline in the ability of the mitochondria, and this is as increased amounts of etomoxer on the x-axis, the amount of fatty acids that can get incorporated into cardiolipin declines pretty significantly. So what this suggests is that the entire pathway, LCHAD may indeed play this critical role in the remodeling step, but because it needs acyl-CoA as its substrate to do so, all of the upstream enzymes from VLCAD all the way up to ACSL1 that are all responsible for trafficking acyl-CoA into the mitochondria and determining chain length, etc., these enzymes all play a role in how cardiolipin is remodeled. So the take-home message on the cardiolipin part of the talk would be that I'd want you to just walk away understanding that cardiolipin is this special phospholipid. It's in the membranes of the mitochondria only, that it anchors these key FAO enzymes, CPT2, VLCAD, and LCHAD, to the membrane and make sure they're in the right place. And lastly, that you need to have a properly functioning FAO pathway to get proper composition of the cardiolipin, and you also need properly functioning cardiolipin to have optimal FAO. So there's a lot of unknowns in this field. We don't know to what extent these changes in cardiolipin contribute to the FAO disease symptoms. And we don't know then, would that make cardiolipin a good target for new therapies? There's some emerging evidence from Jerry Bockley's lab that suggests that indeed cardiolipin might be a good target. When he treated FAOD cell lines with a special peptide that binds to cardiolipin and stabilizes it, that he found improved mitochondrial function. So this will be something to look for in the near future in the scientific literature. So I'd like to turn now and use the last 10 to 15 minutes to talk about reactive oxygen species, or ROS, a lot of people say. Reactive oxygen species are exactly what they sound like. They're molecules containing oxygen that are highly reactive, and they'll react with other molecules in the cell, such as DNA, RNA, proteins, lipid membranes, and when they do so, they damage those components, which can cause, eventually, it causes something we call oxidative stress, if you've seen that term, and eventually, if it's not dealt with, can cause cell death. For mitochondrial biologists, we're particularly interested in one component of ROS called superoxide, which is formed largely, not exclusively, inside the mitochondria through leaks from the electron transport chain. When it's not functioning at its best, or when there's a mismatch between the input of electrons and the output of ATP, electrons may build up, they leak out, they jump onto oxygen. Now you have oxygen that has an extra electron. This electron is unhappy, it wants a friend, a partner. This makes the superoxide angry, and it wants to either donate an electron or steal an electron. And if it's not dealt with, it'll steal an electron from other cellular components, causing oxidative stress. Superoxide is very short-lived. It's being constantly made inside your mitochondria, but it doesn't last long because it either reacts with something or it encounters this enzyme called superoxide dismutase, SOD for short, which converts it to a less damaging form of oxygen called hydrogen peroxide. You may recognize that term, and you may indeed have a bottle of hydrogen peroxide at home. Hydrogen peroxide is not great for your cells, but it's a lot easier to deal with. It can leave the mitochondria where it encounters several enzymes in the cytoplasm of the cells called GPXs or catalase, which can convert it to water and oxygen and get rid of it. Now, some experiments have shown that if you have zero hydrogen peroxide in your cells, that's actually bad. Ideally, you want low levels of hydrogen peroxide. It plays key signaling roles in something we call redox signaling, which is essentially a healthy form of stress, oxidative eustress or good stress. It's only when the hydrogen peroxide accumulates beyond what's thought to be 10 nanomolar, get up into the 100 nanomolar range, you start to run into trouble. It reacts with other things, such as iron, and you get these nasty things like the hydroxyl radical, which can cause a lot of damage to your DNA, membranes, etc. And this is oxidative distress, and if it's not dealt with, it'll cause disease. So some recent papers have shown that FAOD patients, at least with some FAODs, may have higher superoxide production. In this paper from Jerry Vockley's lab from 2 years ago, they looked at VLCAD patient fibroblasts. And what they did was they took away sugar from the cells, which makes them unhappy and stresses them. And they saw that when they do that, you now see this dramatic rise in ROS in the patient cell lines. In this case, they're numbered, fibroblast 671, 773, 833. They all react similarly. If you just compare these last two bars to each other, you're looking at the normal control cells and the VLCAD cells in all three cases, and you see more superoxide and ROS. Other studies have found the same thing. Here's a 2018 paper, looked at several VLCAD patient cell lines. Control is in the white, and the patients are in the gray. And you see all of the patients have a tendency towards less ATP production, signaling dysfunctional mitochondria, energy production. And most of them, but not all, also have more ROS than the control cells. And there's also a paper implicating ROS in MCAD. In this case, again, they used fibroblasts. They saw no differences under normal, unstressed conditions, but when they stressed the cells with a mitochondrial poison called antimycin A, in the red data here, you see that MCAD knockout cells accumulating more ROS than the control cells in the purple. But surprisingly, this doesn't extend to all of the FAODs. In fact, CPT2 deficient muscle cells in culture have lower ROS. For this graph, I want you to ignore the black bars. That's part of a separate experiment in this paper. And just look at the white bars. The control subjects are set to 1, and now you can see about 30% less ROS in each of the 4 patient cell lines. And we don't know why that is, but what it says is this phenomenon does not extend to all of the FAODs. But what can we say about what's going on in a patient, in the patient's body itself? Well, that's really hard to say because we can't really look inside the intact tissues to see what's going on. There are scientists working on technologies that would allow us to image ROS in real time inside of a patient, but we're not there yet. But what we do know from this 2019 paper is that some markers of oxidative stress, such as damaged lipids, damaged DNA, show up in the urine of LCHAD patients, somewhat of MCAD patients, and also multiple acyl-CoA dehydrogenase deficient patients. Particularly the damaged DNA marker is high in both of these, a little bit less so in MChem. So what this suggests is that this oxidative stress is occurring somewhere in the body of FAOD patients. We don't know which tissues or exactly how much. These markers are all just relative to control. So why do the FAODs produce more ROS? Well, one explanation would be going back to these mitochondrial electron transport chain supercomplexes. I said that they're more efficient when they're glommed onto each other in a big happy ball, particularly complexes 1, 3, and 4 of the respiratory chain. And we know from Jerry Vockley's lab that FAOD patients, VLCAD has been studied fairly in-depthly in his lab, have less of these supercomplexes. Instead, they tend to just be spread out as individual complexes. And that's believed to allow for a greater rate of electron leak to oxygen, making more superoxide. But there also may be a role for non-mitochondrial sources of ROS. In this interesting paper, they looked at several LCHAD fibroblast cell lines, 8 of them, and 1 VLCAD cell line. And what they did was they stained the levels of ROS in these cells using a red dye that binds to superoxide. And you see in your control fibroblast cell lines, there isn't any red color, very low ROS. But the LCHAD cells, all of them, and the VLCAD cell line, all have some degree of red staining indicating ROS. And then they hypothesized that some of this could at least be coming from an enzyme called NADPH oxidase. Not all of the cells in your body, but some of the key tissues, particularly in the immune system, have this enzyme on the outside of the cell. And it's there strictly for the purpose of generating superoxide on purpose, because superoxide will kill bacteria. And so this is a defense mechanism to try to stop potential invading bacteria. So what they did was they went back to all their cell lines and they put on an inhibitor called FOX, FPHOX 12, which is known to inhibit this NADPH oxidase, stop the production of superoxide. What they saw in all cases was the red color disappeared. So this is some evidence to suggest that not all of the ROS is coming from the mitochondrial respiratory chain. There may be other mechanisms involved, at least in some cell types. So what can we do about ROS? There's a number of research labs that are trying to develop drugs that may scavenge or help eliminate superoxide. One of these under investigation here at the University of Pittsburgh is called JP4039. Jerry's lab has studied this drug fairly extensively in VLCAD patient cell lines. They published 2 years ago data showing that in 2 out of 3 of the cell lines they looked at, we're going to compare now this third bar to the fourth one, treatment with JP4039 in the patient fibroblasts reduced the amount of superoxide. This patient in the middle, 773, did not respond to the drug, but the other 2 did. So this is another emerging area of investigation to keep your eye on for the future, is the possibility of using scavenging drugs that may reduce ROS. So I want to stop now with just a quick summary of the major overall conclusions of what I've told you today. I hope I convinced you that there's growing experimental evidence to indicate that both cardiolipin and ROS are altered in many patients with FAODs. These may in turn be causing the mitochondria morphology to change, and also may be causing functional changes that we still don't fully understand. These 2 changes, the cardiolipin changes and the ROS might be related to each other, because we know that cardiolipin is the platform for supercomplexes in the mitochondrial membrane, and the supercomplexes are important for minimizing ROS. So that also requires further investigation by basic scientists. The long-term effects of these changes and how they might contribute to the symptoms remains to be determined. We don't understand that yet. And similarly, another active area of investigation is how cardiolipin and ROS might become therapeutic targets for reducing symptoms in FAODs. I will stop there and take questions.
Wow, thank you so much, Dr. Goetzman. I have been viciously taking notes here, and I hope the questions that I have and the questions that our viewers have also I can articulate. The first question I have is, since the heart appears to be picky on what it uses, is this the reason there is so much more cardiac involvement seen in FAOD patients?
We don't understand how much these things contribute, but certainly yes. The heart is unique in that it needs energy all the time. It never stops. So your skeletal muscle, you can kind of control for that and compensate based on, whether you eat before you exercise to keep your glucose levels up, how intensely you exercise. But the heart, you can't really do much about that. It wants energy and it wants it all the time. And that's probably why it wants this very specific form of cardiolipin, would be to maximize its efficiency. So even a few percent loss of efficiency could make a big deal to the heart, whereas it might not for other organs like the liver or the brain. I hope that kind of makes sense.
No, that makes complete sense. We have a comment and a question. This is a great talk. What do you envision might be the effects on a CPT2 deficient mitochondria of that dramatic shift to highly unsaturated cardiolipins?
Yeah, unfortunately, I don't really know. I mean, I could guess, but at this point, nobody's really been able to manipulate cardiolipin to the point where you can, let's say, have a very specific profile. So what kind of needs to be done now is to take these cell lines and really look in depth at the mitochondrial function and try to see how it correlates, these unsaturation, how it might correlate to function. My guess would be that, the more double bonds you introduce, I showed those pictures of the fatty acids where they become kinked or they take on these hairpin structures. It's going to change the shape of the cardiolipin dramatically, which it may be what messes up, for lack of a better term, those cristae, those curved parts in the inner membrane, which really could have some significant effects on the ability of these supercomplexes to find each other and to form properly. So I know that's kind of a non-answer, but I would guess that it does have a pretty significant effect.
No, I thought that was a good answer. And that leads me to a question I wrote down is, do you know why complexes 1, 3, and 4 want to be together and form a super complex? What happened to 2 and 5? Why aren't they included?
Yeah. It's because of the individual functions of those complexes, which I didn't really have time to go into, but complexes 1, 3, and 4 are really sort of the business part of the respiratory chain. They're the ones that are most actively accepting electrons, pumping protons, and passing the electrons down the chain. And complex 5 is also known as ATP synthase, and it can kind of exist on its own. It doesn't need to be connected directly with the others. Its role is to make the ATP. It basically harnesses the energy that the others are making and converts it to ATP.
Okay, that makes sense in my world, so I'm glad to hear that. Do the ROS created in the mitochondria remain in the mitochondria, or where do they go?
Yeah, they do not. The superoxide I mentioned is very short-lived. It's so reactive, and it very quickly gets converted to hydrogen peroxide, which is able to cross the membranes and just leave the mitochondria. It can even leave the cell. So, a lot of scientists are studying cells in a dish, and they can measure the hydrogen peroxide just appearing in the media coming out of the cells. So it's basically able to traverse any membrane that it encounters.
Oh, so is that what would be what we're looking for when our patients are taking some of the antioxidant type medicines to help remove that hydrogen peroxide out, the ROSs out of their cells?
Yeah, exactly. Yeah,
I get that.
antioxidants will help get rid of that.
Okay. Are there things that we could include in the diet that would act as ROS scavengers? I saw a paper titled Nutritional Countermeasures Targeting ROS in Cancer.
The answer would be, I think, probably. I'm not really up on all that literature, but I know there's things like vitamin C and vitamin E that are thought to be good scavengers for reactive oxygen species. So I would think the answer is yes, that you might be able to improve your body's natural defense against these things by what you eat. Similarly with cardiolipin, we still don't really know what the nutritional effects are on cardiolipin, because in the heart, I mentioned the cardiolipin likes linoleic acid, which is something you have to eat. Your body can't make that.
Mm-hmm.
It's called an essential fatty acid. So it's still up in the air. If you eat more linoleic acid or fish oil, would you end up with more unsaturated fatty acids in your cardiolipin? Those are ongoing questions that I wouldn't be surprised if that's the case, but we don't really have the answer yet.
So how do you know? I feel like in your world of study right now, you're looking at the chicken or the egg. How do you know which way to start studying and where to put your emphasis? Which is just learning the effects of the ROS more important, or does everything kind of like a Venn diagram go together with each other, and you have to try to balance it out?
Yeah, I think it's like the latter. They definitely all affect each other. In this case with the FAODs, we know what started it all. It's the mutation in the FAO enzyme, and so now it's a matter of trying to figure out, is it the cardiolipin causing the ROS, or which, like you're saying, it's probably not a simple answer. And so we kind of have to look at all of them. And ideally, we need to single them out one at a time so we can understand their individual contributions to the disorder.
Do you think it would be fair to speculate that people with mitochondrial dysfunction are maybe overlapping more into the FAO world, or vice versa? Or do you think they're going to continue to be separated? Or do you think your research is going to show that there's probably more intersect than originally thought?
I would think definitely more intersect. Yeah, that's something Jerry Vockley is actively studying from the side of the FAODs, just showing that FAODs have secondary dysfunction in the general mitochondrial function in the respiratory chain. And certainly it can go the other way as well. I mean, we know from our experiments that if we inhibit the respiratory chain on the mitochondrial membrane, FAO essentially stops, because you need a functioning respiratory chain in order to be able to continually break down the fatty acids.
Right. I have a child with a complex 3 deficiency, so that's why I'm really honing in onto what you're saying, and I really feel like he's dancing on the line of being more in the FAO world than where we currently think he's at. So this is
Yeah.
very, very interesting to me, and I'm sure many of our viewers as well. So,
Yeah.
well, those are all the questions I have in my chat box here. So I just want to thank you so much for giving us all this information. I have a hunch this presentation is going to be listened to numerous times over and over because there's a lot of great information in there. And I just thank you so much for your dedication and your persistence in digging into this for us.
I hope it wasn't too much information.
Oh, no, not at all. No, not at all. I would say it's fair to say it. We will be having you back for more questions. That's for sure.
Okay.
So up next, it's time to head to the lounge for our roundtable discussions. At 12:30, we'll be back in Auditorium 1 for 30 minutes of family yoga. So we'll see you back here in Auditorium 1 at 12:30. And make sure you head over to our exhibition hall to spend some time with our exhibitors and to have some fun in the photo booth.